Hi, the following content is for educational purposes only and does not constitute medical advice. If you are a healthcare provider, please consult with your affiliated institution or local ethics committee for ethical guidance related to clinical practice.
Hello, bioengineering ethics enthusiasts. Today we have a CRISPR update with Dr. Magnus. I'm Russ Altman, as always, and I'm just going to put on the share and then we'll get started. One moment, please. Share, share, share. Come on, share. You must be here somewhere. Share and share.
Sounds like you're talking about a singer.
Yeah. Oh, well, they don't know who that is, but yes. Okay, so
She was
this
on
is,
Saturday Night Live this season.
did she?
Yep.
All right. CRISPR. CRISPR update. We'll call it an update and let's put a date on it, 2026.
There we go.
Okay, what do we got?
Okay, first update is just to update you on the ongoing, the days of our lives applied to Dr. Zhanghui He, our favorite CRISPR, germline CRISPR person who went to prison in China for 3 years, and China was very clear about those changing their laws so that it's now illegal there, as it is in almost all of the world.
Did he
To,
learn his lesson, David?
I would say not. I would say, he got released from prison in 2022. He tells us that the three babies are healthy. Maybe that's true. I have no idea whether
This
he's
is based on a Wired article, which we will
this
make
based,
available.
yeah, Wired article. A Wired interview.
Right.
He says the three babies are healthy, but I don't think he's a very reliable reporter. He says he wants to do more CRISPRing of embryos, but it's illegal virtually everywhere, which is a problem. He says lots of people are coming to him and giving him lots of money to do research, and he's doing some basic research and prep for trying to go back to doing CRISPR babies. He's doing a lot of social media that details the, account of his dating and failed relationship with another media person, social media person. You can't make this stuff up, right? So, he
He is
insists.
positive that he's a trailblazer who will be remembered as a great innovator.
Yeah, it's amazing. As a condition of Wired being able to interview him, he insisted that the article had to describe him as, quote, a pioneer of gene editing, but he has colorfully referred to himself on X as Chinese Darwin, Oppenheimer in China, and China's Frankenstein. And he often posts photos of himself in a crisp lab coat posing alone near scientific equipment. One glaringly empty lab, they say, this is from the article, comes with the text, I did not violate ethics, I overturned it. And that's what we always do to people who overturn ethics. We send them to prison for 3 years. Although we did, in fairness, Socrates was put to death. But anyway, more recently, he dropped the austere look and posted an image of himself seated on a giant throne with prehistoric animals at his feet, a rainbow beaming down on his crown, and a double helix adorning his purple robe. So that's also from this Wired article. So I think it's safe to say that he has a healthy ego. I think that's a fair thing to say. And that 3 years in prison did nothing to detract from that.
Okay, and then believe it or not, that's not even the topic of today's. That's
No,
that's just a bonus update.
that's a bonus update. The real thing is, since then, there's actually been some real science. Dr. He is famous for not really publishing articles, which has continued. He's still not publishing anything in the peer-reviewed literature. But now some major developments really have taken place. That's very much worth talking about and highlighting. So, let me start with this by giving you guys a little bit of background, though, because I don't think we've talked very much about gene therapy. I don't think we've really talked about that
Not too
anyway.
much, no.
So, just a very quick, brief history of gene therapy, sometimes referred to maybe more accurately as gene transfer research. Gene therapy does suggest that it's therapeutic, which isn't always true. In the 1990s, there was a lot of hype about gene therapy, even though, again, no therapies for many, many, many years, decades. Um, Lots of hype, no success or limited success in actually treating anybody with any of these gene transfer interventions that were under investigation. But there was so much hype, and I will confess, Russ, I bought into the hype. I published a paper predicting in the 90s that all cancers were going to be cured and we were going to change the concept of genetic disease because gene therapy was going to revolutionize how we treat things. And I was convinced that the HSTK GCV system, which we don't have to go into detail about what it is, was
David, you
going
were
to
young.
change
You,
things.
you were young.
I was young back
You were
then.
almost as young as the people who were watching this video.
I wasn't that young. I was already a professor,
All right.
but I did have a lot more hair. So, um, so 1997 and following, there was a major setback for the field when what Time magazine described as the high-tech death of Jesse Gelsinger took place. Jesse Gelsinger was an 18-year-old young man suffering from OTC deficiency with an adenoviral vector developed at the University of Pennsylvania by James Wilson. They gave him a big bolus of a properly functioning OTC gene. And he wound up having a terrible reaction and dying. I was actually at Penn at the time, not at the time he died, but shortly afterwards, like within a few months afterwards, and wound up being called on to go with the team before the recombinant DNA advisory committee on that. It was very interesting. One of the big problems that's going to come up that we'll talk about later was once it came out that Jim Wilson owned huge amounts of stock in a company called Genovo that was sponsoring this research and that stood to make he stood to make a huge amount of money potentially by the success of gene therapy. In addition to his very driven ambition, as was true for most of these early pioneers in gene transfer research, of he wanting the Nobel Prize and all that kind of stuff, he was very conflicted. And so was Penn. Penn also had an intellectual stake in this company. And so for this reason, they were really crucified in the media, destroyed by FDA and a huge setback. And he wound up salvaging his career later by talking about like sort of I was there, sort of the Watergate convicted people made career writing books about like, oh, how wrong I was, and now I've seen the light. And that was sort of a lot of Wilson's later career, though he did go back to science eventually. In the 2020s, gene therapy started to move forward.
I just want to point out that that's 23 years of being in the penalty box. That's a long time for, for going from hype to like a wasteland.
Yeah, that's exactly right. There was no money that became a ghost town, right? The gene therapy
Right.
world for the most part. But the 2020s started to have some success, and then CRISPR came along and gave it a big boost. And the development that we want to talk about today is something that happened just in the past year and is continuing to ongo, which is in 2025, a big CRISPR development to actually treat somebody um, using essentially a CRISPR-based gene therapy. Okay, so this is the big development. And the big development is named KJ Muldoon.
Which is a great name.
This is a great name for a cute little
This is
kid.
a little
Great
baby, right?
little baby, cute little baby.
Okay.
At birth, he was diagnosed with carbamoyl phosphate synthetase 1 deficiency, CPS1.
So wait a
Uh,
minute, let's get that right. Oh,
CPS1.
I got it. Carbon. It's probably carbam.
Carbamoyl, is it, it's an O?
I'm gonna go ahead and spell it like this because I used to work
I
on a
think it's an O, it's carbomoyl.
All right, the bonus points for whoever figures that out.
I'm pretty sure it's Carbomoyl.
The reason I don't think so is I wrote a paper when I was an undergrad on carbamyl tri on an enzyme that I think is that molecule, but I could be wrong. Let's just call it CPS1, which
And more importantly, in some ways, it's a urea cycle disorder
that's right.
or UCD. And there are a lot of, it's a rare disorder, but there are a whole bunch of rare urea cycle disorders, not just one. There's a ton of these. The disease that Gelsinger had was a different one. So there are a lot of these rare urea cycle disorders. And with this rare urea cycle disorder, as with most of them, protein, especially and things that you normally eat in a human diet that is
Right.
necessary for development, produces a large amount of ammonia that builds up in the bloodstream. And as a result, about 50% of the infants with CPS1 die in infancy, and those who don't have major neurological impairment in the limited number who wind up surviving. Okay,
Brain.
so it's a
You
terrible,
said brain
profound
impairment.
development, profound brain injury, profound developmental delay.
Okay. So
Um,
not a good disease at all.
Not a good disease at all. The only treatment for CPS1 is a liver transplant. Unfortunately, you can't transplant an infant until it reaches a certain weight. I think it's 14 kilograms, which usually doesn't happen until they're about 7 months old. Which means during that time, there's a buildup, there's risks of dying up before then, although most of the time it's later than that. There is potentially neurological damage that happens. There are some drugs that they go on that helps reduce the impact, and there are diets low in protein and low in things that they wind up being put on to try and reduce the damage until they can do a liver transplant, and then presumably they have to continue with this medication and this altered diet potentially forever. Well, or until there's a transplant. Once they get the transplant, hopefully that will fix the problem.
And the transplant is pretty much curative because that liver has a normal enzyme.
Correct. And so, right then they have this new disease of having a liver transplant,
Right.
which means in
Right.
lifetime immunosuppression and all the problems associated with that. Presumably, they'll at some point have to have another liver transplant because they don't last forever and all the other problems associated with it. Okay, so that's kind of the background, and that's the disease that KJ Muldoon has, or had. Um, at the Children's Hospital of Philadelphia, which is associated with Penn, which is one of the two top children's hospital in the country. There's always a battle between CHOP and Boston Children's. Um, they developed an innovative approach that they wanted to take towards I want to call it treating. They wanted to develop, and it's hard to say the right word for this. They wanted to do a research project that was kind of on the bubble between research and treatment, where they would deliver the precise genes responsible for his particular version. So they sequenced the parents, they sequenced him, and they knew exactly what sequences needed fixing. And then using that and using CRISPR and delivering it through nanoparticles, they were able to deliver a precise, targeted CRISPR treatment to cells in the liver that will allow KJ to start producing the correct enzymes.
So in our other modules on CRISPR, we discussed actually how it works. So they must have given like guide RNAs and
That's
all that
exactly what they
stuff so
did.
that it could go in and it could actually edit the genome or other DNA and put in a new version or fix the old version.
That's exactly what they did. And
Okay.
I don't think they like transduced like most of the cells. I don't think they edited most of the cells, but if
Right.
they edit enough cells so enough enzyme gets produced, a lot that's proper, which happens with a lot of these kinds of urea cycle disorders, it doesn't take a lot to wind up having sometimes a big impact.
That's right.
Um,
That's the really good news is that as long as some of these cells are making the gene, they can metabolize the ammonia and not have the high levels.
So far, so good. He's had reduced requirements for drugs, and he's been able to eat at least some protein, which has helped him in meeting milestones of development as he's starting to age. So that's all really good news. Um, but that's really what's going on with this treatment.
So I
Now,
want to point out that you made a big appropriate deal about the fact that you said you weren't sure you would even call it therapy, it was research. So these beneficial effects are not really what they promised the parents or perhaps even what they expected. Or is that
well,
not
that's what they were hoping for. The question of what they told the parents and what the parents expected we will get to. So you're
all right.
jumping ahead a little. But
All
that's
right.
a big issue.
Okay.
Okay. So and the reason why this matters, you might say, well, it's a rare disease. So, what's the big deal? So this is a one-off. There aren't a lot of people with CPS1. But in fact, one of the things that's interesting is that these rare diseases like urea cycle disorders, but other rare diseases, even though they're individually very rare, the collective impact of all of them is very large. So there are 7,000 rare diseases that are afflict people in the United States, at least there's 7,000, probably more, and that affect at least 30 million people in the United States and 300 million people worldwide.
Wow.
What this means, and what's interesting about this treatment is it's individualized, right? So and we'll talk a little bit later what that means, and why that's significant. But this is like his specific sequence that needed to be fixed. Other kids with other similar diseases, it'll be a different sequence that will be fixed. And that means that both there's a potential for individualized CRISPR-based therapeutics for potentially thousands of patients, maybe tens of thousands of patients, maybe hundreds of thousands of patients, depending on how severe the illnesses are, these rare diseases are, and whether they're good targets. And there's a lot of questions to be raised about how many people will benefit from this and how much, and whether the risks are worth it. So there's a lot of issues here, but it's potentially a large number of people that may eventually. So that's one of the reasons why this is getting so much play is people are seeing this as the beginning of the future of medicine, much like we did earlier in the 1990s, but maybe this time it really is the beginning of the future of medicine. It's certainly a lot of excitement generated about this treatment, this intervention. I should call it an intervention. This
There might be a
intervention.
young David Magnus who's getting overexcited about the potential hype here.
Yeah, maybe. Oh, but maybe they're not. Oh, maybe it's not overhyped. And I will say right now, lots of places around the country, including Stanford, are considering getting in the game. So lots of right. So we did a very extensive research ethics consult with the teams here who are working on this, and they're working in collaborations with folks at Berkeley to do this. But there are teams around the country who are all racing to get into this game. CHOP is moving forward with other interventions. So it's fast moving right now in this space. And the ethics of this, there's not much there. We have an article under review at Cell that looks promising. So that would be the first real ethics piece on this new approach for CRISPRing babies the right way, not through the germline, but
Right.
somatically CRISPRing babies.
That's an
And then
important. Let's highlight that. This is a what we call a somatic gene editing, which is not the germline. This will not be passed on. If those babies do very well and have lives where they're able to have children, those children are still at risk depending on the genetics of the disease. This is not going to fix it for all future generations. That's what we mean by somatic non-germline.
that's right. And that's right. Um, in addition to urea cycle disorders, which are sort of the obvious first target, there's active research on trying to CRISPR to some of these rare diseases to target bone marrow, T cells, and brain cells.
Wow. So yeah.
Pretty
Now,
exciting.
the liver, yeah, the liver, I should say, is a little bit easier because we have viruses that are extremely good at seeking out the liver and infecting. Maybe not 100%, but 10, 15, 30%. I don't know about brain cells, for example.
But they're not really using, I don't think they're using a virus as a vector this time. They're
Yeah,
using like nanoparticles.
that's probably is that true for this one too?
That's for this one. For
Okay,
Gelsior, they
so
use an adenoviral vector.
right.
For this one, they're using, which could, for all the reasons you said, viruses are very good at transducing cells in the liver. But this one, they're using I think they're using nanoparticles to deliver the CRISPR and the guide into the cells and into the.
Yes, yes. So that's interesting, and I don't think I've done a technical briefing on that. But most of these nanoparticles are such that they can fuse with a cell and basically dump their cargo into the cytoplasm of the cell. That's the mini technical update.
There we go.
Okay.
Okay, now the regulatory situation here is very complex.
Yeah.
So on the one hand, these are all individually very rare diseases, and the treatments are very individualized. And what that means is the typical pathway for drug development or biologic development will not work, right? The normal situation that we normally go through in drug development, which I don't know if we have this in any videos. I don't think we
No,
actually
I
do.
don't think we've talked about drug
But
development.
normally when you're developing a drug or a biologic, you start with what's called a phase one trial. And in a phase one trial, you get a small number, relatively small number, sometimes as few as 8 or 10, sometimes more like 20 or 30 of individuals, usually for adults starting at very low doses. For kids, you don't do it quite the same way. Like for KJ, they started at a very low dose and they didn't see any impact, and so they gave more doses until they started to see an impact. Um, But so anyway, so you start sort of slowly and build up, and you're really in phase one, really just trying to determine the maximum tolerable dosage and see, and it's sometimes referred to as a safety study. In phase two, you're sort of continuing the safety evaluation, also with a small number of research participants. But you're in a phase two clinical trial, you're starting to look to see whether it looks like there's some signal there to suggest that it might work. You're not establishing that it works or proving that it works. You're just trying to establish that it's plausible that it might actually work. And then in phase three, that's when you do the randomized controlled trial, right? That's when you do that's the full-blown randomized controlled clinical trial, where also known as RCTs. Sometimes those are placebo-controlled trials, sometimes those are active controlled trials, but that's where you have to have them powered sufficiently and statistically to be able to establish benefit. None of those phases are plausible or possible with these kinds of interventions under investigation, right? There aren't ever going to be enough people with these diseases to do a phase three trial. And unlike the earlier gene therapy, like when they were doing the work research on Jesse Gelsinger, his trial was phase one, and they were planning on doing originally 18 and then expanded it to they were planning on I think they did 20 people in that one. But they all got the same thing. They all got the exact same intervention. Here, each patient is going to get something different. So, right? So it's going to be really complicated. These are all N of one trials, is what they're sometimes referred to as. And it's a struggle. It's historically been a struggle in our regulatory system to think about those kinds of things. Because if for each and every genetic disease, or each and every, even worse, even every unique allele or set of alleles that they're going to wind up, or sequences that they're going to wind up pushing in, if they have to go through a full FDA approval process for each one of those, it's going to take a very too long to ever get
Yeah.
any of these things approved in a way that's very
And
timely.
you'll never get statistics, which is what the FDA likes to see.
Yeah, and that's never going to happen for these things. So initially, my understanding is that this was approved as an N of one expanded access trial, which is really an amazing development as an approach. Expanded access, sometimes called compassionate use, that's something that arises when somebody has something that they think might be work. Usually, the requirements for this are usually that they're already through phase one. But sometimes, if somebody has a disease, like let's say it's very similar to another disease, but it's not the same, so they can't be enrolled in the clinical trial going on, but it's somewhat promising and there's nothing else that works, they will sometimes be allowed to get compassionate use or expanded access. And so, anyway, but that's one way of getting approval. Another way of getting approval, which I think the FDA is working on, is a change that took place in 2022, where the FDA introduced the idea of a platform technology where you don't approve all the details, but you approve the overall platform. So the idea behind this would be they would approve, the CRISPR technology, the guide RNA. They would approve the methodology for doing this because that's going to be the same, and all that would change would be the actual sequence that's being introduced. And then maybe it could get approval as a platform approval, and then it wouldn't matter that each thing you're doing is individualized and slightly different. But in any case, this has been a real challenge from a regulatory point of view, with the FDA indicating that one way or another, whether it's compassionate use or slash expanded access or platform technology approval, that they are trying to find pathways that will allow this to happen much faster so that they can allow these interventions to happen earlier in the process. So that's kind of but it makes it very hard to learn from these things because
Right.
you're looking at each one individually and saying, like, did something disastrously bad happen? And if it did, was it from the disease or from the intervention? Going to be
Right,
very hard
exactly.
to
So when
tell.
you get a bad effect, do you blame the platform, which is very bad? That means you have to stop using it. Or do you say, no, that was the specifics, which is bad for the patient, but it keeps the platform approved?
Yeah. Anyway, so all of that is going to make it very complicated, and efficacy is going to be also very challenging to establish. I mean, in a way, these are sort of Hail Marys. They're saying like, oh, more protein, reduction in the amount of medicine, but that's pretty weak tea from an evidentiary point of view on an N of one. So, in the ideal world, some of these kids will be cured and no longer need liver transplants. And it's kind of, you could think of these almost as home run trials. It's
Right.
either going to be, in phase three trials, you can see a lot of singles and walks.
Right.
Baseball season is almost here, so
Right.
I've got baseball on the mind. So you can see singles, you can see walks. This is the kind of trial that I think of as a home run trial. It's a home run or it's nothing. It's a singles and out, a doubles and out. It's got to be a home run or it's nothing. Anyway, maybe. Anyway,
Right.
but they're going to try and see what they can learn from these things. There are precedents for that kind of thing at the very cutting edge with rare things where you're trying just to see with a very small number where you might be trying just one or two or three. And I think the way they're working at CHOP, the way we're thinking about working at Stanford, the way everybody's thinking of doing this, it's like we're going to start with one and see how it goes. If it goes well, maybe we'll do one or two more. We'll probably, if we do two more, we'll probably do another one and stop, and then maybe do another one. And then down the road, as information comes in with this platform, then maybe we'll start to do some larger numbers. But this is really challenging because this way of thinking about things and the way this is being proceeding really represents a lot of ethically very challenging blurred boundaries, right? Since normal clinical trials are impossible, and we have to learn from each individual case. That is so much more like surgical innovation. In fact, people have been suggesting that we should think of this, even though it's a biologic, we should think of this not like a drug or a biologic, but think of this almost more like a surgical innovation. Because in surgery, this is what happens all the time. Surgeons try things and see if it was a little better. They like, hey, here's an idea. Let's just do this. Hey, it seems like it worked. Let's try it again. Hey, it seemed like it works. Let's try it again. Hey, it seems like it works. Hey, I'm going to publish my three cases, and now everybody knows to do this instead, which sometimes turns out to be true and sometimes not. But anyway, but that's kind of how surgical innovation goes. Um, it also creates a blurred boundary between the roles, right? So the researchers include folks in these on these teams. You have to have folks on these teams who are involved in the clinical care of these patients. So the role between physician and clinician is very blurred in this
You
kind of
mean
approach.
researcher and
Yeah,
clinician.
sorry, researcher and clinician. So those boundaries are very blurred and make it very tricky because your obligations are different. As a researcher, your fiduciary obligation is to research, and you only have ancillary duties to the person, the research participant. Not that you have no duties, but they're ancillary to your primary obligation as a researcher. You're right. That's why when somebody's in a phase one trial, you don't say, "Oh, for adults, you usually start at very low doses." You don't say, well, we have to start at low doses for this patient, but it's probably not going to be effective because we're just doing a safety study. But this guy, he's got two kids. I really want to help him. So even though I'm supposed to only give him like a thousandth of the dose of what we think will be work, I'm going to give him the full dose in my first cohort in phase one. You're not allowed to do that. You don't individuate that way. You don't get to say in a placebo-controlled trial, well, I don't want to give this guy a placebo, so I'm going to make sure that my patients get it right. Then you wouldn't have a trial. So
Right.
anyway, so the obligation, whereas in clinical care, your primary duty is to the patient.
Yeah,
And
it's
you may
whatever it
learn
takes.
something from that, but your fiduciary obligation and duty is to the patient. It's not clear what the duties are here. They are both trying to produce general knowledge and they're also trying to help these individual patients. So this is a very, very blurred area.
Yeah, and it's probably best to have those two functions in different people. It would be very difficult to be both the patient's doctor and the person who's doing the research trial. They at
It's
least, I would
it,
guess, they at the
yeah,
least need to make them be different people.
they can't they can't every one of these teams has to have the people involved do it. It's just not
Yeah,
feasible
yeah.
without that.
So
And
that's why it's hard.
that's why it's hard.
That's why it's a blurred boundary.
And it creates this ethical tension between these roles and the duties of these roles between researcher and clinician. And I'll just say what makes it even more acute is the intense feeling that clinicians have of the duty, sometimes called the rule of rescue or the duty to rescue. They want to rescue their patients. So I think it's okay if I share this anecdote, but like we have been consulting quite extensively working on this paper with the team who's thinking of doing this at Stanford. We all had agreed on some inclusion criteria. We've been going through all this stuff, and we want to be really careful. It's going to be the first one at Stanford. We want to be really careful about what's involved. And then, of course, what happens is a recycle disorder patient comes over the transom and gets diagnosed at birth. It's like, can we do it? Can we do it now? Like,
Yeah.
we want to go forward now. It's like, well, you got to take a step back. Like as a clinician, I understand. It's like I want to grab. I got nothing else. I want to try anything I can to help my patient, but that's not what the role of the researcher is, and we have to separate those.
But what is this duty to rescue?
I mean, ultimately, so rule of rescue or the duty to rescue
Yeah,
is
what is
that
that?
it's this the duty of clinicians to really do what it takes to save their patients.
Ah, okay.
If their patients are salvageable and it's in accord with their values and wishes, you don't want to salvage patients who don't want to, whose quality
Right,
of life
right,
is
right,
low, they
right,
don't
right.
want to be alive. But this rule of rescue is very powerful in clinical ethics, right? Because like when we give livers to transplant recipients, we don't do it on the basis of who's most likely to benefit. We give it to the people who are the sickest,
Yeah,
even though they may be so sick that they are less likely to benefit from a less sick patient, but we give it to them because we have to rescue them. And I've been
okay.
involved in cases where we have like acute blood shortages. You got a patient in front of you, you're going to empty the blood bank completely. This was a long time ago, but a long time ago, we had a patient with, liver damage uses a lot of blood, and liver transplants do. So you had a patient who was just losing like so much blood, and it just happened to be at a time when the Bay Area was short of oneg, and it's like we were running out of oneg, but like they're like, you can't stop on my patient. If I have to use every bit of oneg in the Bay Area to save my patient, we're gonna do it. And it meant that we had to shut down our ED, right? So all the pregnant women coming in to deliver had to get diverted to other hospitals because of a blood shortage.
Very
Probably not from a utilitarian point of view, a very utilitarian way of doing things. But so the rule of rescue, it's a deontological fiduciary obligation that often runs afoul of any kind of utilitarian considerations.
all right. Got it.
Okay. Let's see. Other ethical tensions, conflict of interest and conflict of commitment issues. We want to make sure that there's no repeat of the Jesse Gelsinger experience. But that's also going to be hard because ultimately this stuff's going to be expensive. The research is going to be expensive. Who's going to pay for the research? Anyway, so the risk of significant COI and
Yeah,
COC.
and you're going to want the people who do this best to start a company to bring it to everybody else, but that immediately creates a risk, a
That's
conflict.
what happened to Wilson and to
Yeah.
Penn, right?
And that's an important point to make. It is not ridiculous for these people to start companies because they become literally the world's expert about how to get these technologies to work. So in other ways, they're exactly who you would want to start the company, but problem. Okay.
And then another ethical tension is around therapeutic misconception and misestimation, which is what you brought up earlier, right?
Yeah.
Right. So we know that this is already a big problem that participants in research often overestimate um, how likely it is that they are to benefit. And this is a kind of research where both the risks and the benefits are completely unknown. We don't really know if this is going to work, and we don't know, you know, these kids are so badly off, it's hard to make them much worse off, but they could die instantly, right? So
Right.
they could be a little bit worse off. And it depends on who's in it, right? If you're doing a urea cycle disorder like this one, that's different. But what about trying something for macular degeneration? What about, as the people try different things for diseases that are less serious, the amount of risk, the benefit risk ratio starts to change and is relevant for thinking through who ought to be a participant in this. Anyway, and so people have a tendency to misestimate, and then the therapeutic misconception arises when people misunderstand what this is. Is this right? So you look at in the early gene transfer research, which is often called gene therapy, they all thought they were getting therapy when they were actually in a phase one trial. And
Right.
right, and the goal is just to find maximum tolerable dose. So you want to make sure, and that's true. In phase three, Paul Applebaum has done studies where patients in placebo-controlled trials believe that they're getting individuated therapy and that they're deciding that I'm not going to give the placebo because my doctor knows I'm old and I don't have that long to live and I need this. And so I'm not getting a placebo. So they've confused what the role is of the person. And again, the fact that it might be a clinician involved as well as other researchers. So even if you can't get rid of the clinicians involved, it does suggest that the people actually recruiting them need to be CRCs or people who are not the actual recruitment and informed consent process needs to be separate and probably good idea in the early phases to have a patient advocate being involved in this. So here are some
Good.
of the ethical issues that need to be addressed for this to go forward in an ethical fashion. First of all, there's a whole range of issues around participant selection. I should say condition and participant selection. Whoop.
Sorry, we're good. So, I'm sorry, I was catching up. Ethical issues.
Ethical issues to be addressed. Okay,
Good.
that need to be addressed for this to be able to move forward. First, issues around condition and participant selection. You got to
Yeah.
choose the right condition. How serious the disease? Urea cycle disorders, I think, are a really, really good first target.
Uh-huh.
Um, uh, what other existing clinical alternatives exist? We've already seen the alternative is a transplant if it's available at 7 months of age. Um, there's also going to have to be some equity considerations as we think of the participant selection, right? Is this going to likely be given mostly to wealthy people in the early days and as it starts to develop, or is this going to be equitably distributed, especially when it's not being paid for? This is all going to be stuff done on spec by the institution. And then there might be consent considerations that are relevant to the ethical issues, the choice of participants. And I'll get to that in a little bit when we talk about informed consent. Okay, so then that other issue around informed consent, want to avoid misconceptions. We've made some suggestions for that, like having a patient advocate, not don't have their clinician be the one consenting them. Here's the other issue that's relevant to both participant selection and informed consent. Who should the first participants be? Like KJ was diagnosed at birth, shortly after birth, when they saw that something was going wrong. Think about this from the perspective of the parents. You thought you're having a healthy baby boy, you just gave birth to him, and now something's terribly wrong, and you get this horribly grim prognosis and told, we want to try something new and innovative to see if it'll help. Is consent even possible under those kinds of circumstances? Maybe it is. We certainly say that it is when we ask parents other things like, should we intubate or not? Should we go forward with a risky, surgery with a small chance of success? So they certainly consent on the clinical side to lots of things in those circumstances, but it's certainly far from ideal from what you would want to do. So a lot of times these urea cycle disorders are diagnosed in utero. So maybe the very first ones should be done on at least the first one or two or three should be done on patients, sorry, who have been diagnosed in utero as having these urea cycle disorders. The only challenging thing there is you don't know how bad it's going to be yet sometimes. You don't know exactly what you're going to see.
Is this to give the parents more time to consider?
Correct. So you can talk to them about it and say, we're worried this might happen. We're not sure, but there's a good chance that this could be happen. And if it does, here's what that's going to look like. If it looks clinically like that's the setting, here's an alternative research thing that we're trying to do and that we want you to think about. And you have a little bit of time, you have a month to think about it. And then after the child is born, then you can start to see the clinical manifestations and talk to the family some more after they've had time to really digest all this information and think about it. So you might get a better informed consent process, and maybe using better informed consent process as a way of selecting at least your first few patients might make a lot of sense rather than taking whoever comes along. There are two problems with that, right? One is the people who are likely to have been diagnosed in utero are more likely to be resourced people, which leads to a less equitable selection, although it will only be for one or two or three people, presumably. And then second, there is, that rule of rescue is going to be very challenging when you know you're going forward and a kid is diagnosed at birth with this thing, saying, I'm sorry, we've decided the first three are all going to be ones who were diagnosed in utero. It's going to be very, very hard for the clinicians involved to know that that's sitting in their back pocket and they aren't going to be able to use it. But that's the nature of research, right? It hasn't been established as it's not a proven treatment. Right now,
Right.
we're in equipoise, if you like to think of it that way. We don't really know whether or not this is going to work or make things worse. Okay, another issue is going to be the need for long-term care and monitoring. And then, given the nature of the fact that these are going to be either um, you know, uh, um, compassionate use slash expanded access or platform approvals, the amount it means the amount of IRB oversight and the amount of FDA oversight is not, let's call it, as robust as you would normally expect for a new drug trial or a new biological being introduced. And that suggests that maybe this is something that's in on the Cell article that we've submitted. There's a need for institutional meso-level process guidance. That's you need something between regulations like the FDA and the IRB and the bedside. The institution needs to step up and start to develop processes. And I will say our researchers have been very responsive to the idea of doing that and using things like patient advisory boards and having ethicists consultation as part of the process, so that you get to hear from parents. You get to all this institutional feedback on this as you're thinking of going forward. But a lot more needs to be done in that space. Okay, another issue that we actually didn't address in our cell article, downstream financial implications. Everybody hates it when ethicists talk about this, when people are developing new technologies. When Spinraza, it was the first sort of gene therapy like treatment for SMA1. It was an antisense technology. So when the antisense technology Spinraza for SMA1 was first developed, um, we talked about like how much is this going to cost, what's it going to mean, and all the research people who were like developing this were like, yeah, who cares, who cares, who cares, and of course it instantly became the most expensive drug in the world. And since this was something that was expected to be needed for life, it's going to cost, I don't know, well over $1 million the first year and something close to $1 million a year for life thereafter for these kids
For this treatment?
for the SMA1 treatment.
Yeah, no, but not we don't even know for this
We
one.
don't even know,
But
but it
because
seems
it's curative
likely it's
and yeah, the arguments they make for these blockbuster drugs that are for these terrible diseases is they do a calculation on how much money they're saving the family and society for the next 50 years, and
yeah.
or avoiding death, and they come up with these very big numbers.
Well, $1 million a year for Spinraza, that was not a very good. I mean, if you think of a value for money, right, that's not
Right,
great.
right.
Then a gene therapy came along, so it was presumably just one treatment, and then that's a better bet, even though the price tag was still $7 million. The challenging thing here is ironic, which is as a one-off, everyone was worried, will insurance companies cover the cost of Spinraza?
Right,
Will they cover these $7 million gene therapy? And they were rare, and so the company said, yeah, sure, we'll do that. It's rare. This
right.
urea cycle disorder, sure, if it turns out to work and it looks plausible and you cross some threshold, you still have the financial problem of coming up with the money to get to the point where it's approved and how you're going to come up with that money in an ethical way. But at some point, if it is, it's going to be very expensive. But the problem is what we said before, which is there are a lot of these rare diseases,
Yeah,
and it's the first few are going to get covered. But what are we going to do when it's 10,000 people times 7 million? I'm
yeah.
not sure I can count that high. That's 70 million. Is that how much is that? 7 million times
I
10,000.
never do math during a lecture.
Okay. I can't even think how high that number is.
It's a big, big number.
It was if they charged $100, it would be 700 million. If it was $1,000, it would be 7 billion. If they charge $10,000, it'll be 70 billion. And if they charge $100,000, it'll be $700 billion, which is which, we can't afford that. We just can't afford that. And so this is going to be, so we're sort of in the early stages of a revolution that we may not be able to afford.
Yeah.
So success may turn into failure. So that's a very big downstream implication that I think we need to start to think about now. Although, as I said, it's not in the cell article. Um, then I think there's the ethical frameworks that you have already learned about in these earlier videos for how to think about this. Consider this from the perspective of the requirements for innovation and for general research requirements, right? For innovation, scientific background, institutional field strength, the ethical climate of the institution, professional and public discussion, everything that was absent from Dr. Hu's CRISPR efforts, right?
We're closing the circle now.
Yeah. And then research requirements. Risks have to be reasonable in relation to anticipated benefits, and they need to be minimized as much as possible, consistent with sound research design. You have to have informed consent of the participants and equitable subject selection. All of those are going to be required for this to go forward ethically and to assess this. And it seems like figuring out whether or not those are applicable to this technology might be a good case study for class.
Yeah. I think I hope people watch this and think about it because I think we're gonna bring this to them for future discussion. I just want to get the last point here, which was equitable subject selection.
Yeah.
Well, there you go. There is your CRISPR update 2026. Dr. Ha is back in business. So are a lot of other people. And thank you very much, and we'll see you at the discussions. Signing off. One moment. Stop recording.